Let's go on a trip. We start in lecture where my professor tries to teach her half-awake students. Afterwards, I begin to make my trek home and head to the subway. Before I can access the subway, I have to use a Metro card. It's $2.75 a fare, and they're not that difficult to obtain, and are extremely common in the New York City area. Once I'm on the subway, I eventually make a transfer to the Express, where I simply walk across the platform and head to the Long Island Railroad. In order to board and stay on the LIRR, I need to have a ticket that's specific to the station I'm heading to. These tickets are more expensive than the subway fare and have a more limited use. Once I get to the station, I take the bus. From the bus stop, I walk to my house. As I finally get to my house, I use a key only my family and I have access to. Throughout this trip, we've used multiple forms of transportation, requiring different levels of security and accessibility. Like our own bodies, which carries a similar method of transportation in order for our cells to communicate. I'll transfer trains by just walking across the platform. Cells that are next to or near to each other will communicate through simple messages and or bridges. I'm Antonio Lopez, and let's discuss cell communication. A common bridging method between neighboring cells is called gap junctions. They're made of transmembrane proteins called connexins and form channels that connect the cytoplasms of neighboring cells. Like our train hopping earlier, hydrophilic molecules such as calcium ions simply pass through the channel. Calcium ions can help with potassium levels within cells by increasing potassium conductance. But calcium ions can also be used in our immune system, where they act as an intermediate messenger for the production of interleukin in T cells. A complexer form of cell-to-cell communication is the Notch signaling pathway. It begins with outer membrane signals and receptors that are specific to each other, like a key to its lock. Once the notch is initiated, a signal is sent into the nucleus, where protein synthesis begins. Once the message reaches its target gene, it removes the repressor from the binding site so the gene can be transcribed. Once the transcription factors bind to the gene, transcription can begin. The messenger RNA is built from the DNA molecule, and it is modified so that it can exit the nucleus and enter the cytoplasm of the cell. Once the messenger RNA enters the cytoplasm, ribosomes attach themselves to the RNA and begin constructing a polypeptide chain that will eventually form into a protein. At the end of the Notch signaling pathway, a protein is produced that can be used as a messenger to regulate the phenotype of an organism. In Figure A, we can see the ultimate goal of Notch signaling of developing organisms is limited expression of the specialized cells. Figure B shows when Notch signaling is defective. To the left is when it's off completely, so no cells become specialized. To the right is when it's continuously on and all the cells are expressed. Figure C shows a wild-type organism where its Notch signaling functioned correctly during development. Figure D shows what happens when Notch signaling is defective and how it can drastically change the phenotype of the organism. Our body's own complicated system of communicating can be used against us. Certain viruses can imitate the shape of certain membrane signals of cells and enter into the nucleus. Some viruses can just directly inject its genetic information into the cell. The virus then uses our protein synthesis and DNA replication methods to make copies of itself, thus making our own cells factories for the viruses. But luckily, we can combat against viral infection through our immune system, where this hijacking of cell communication can be put to an end and restore it back to our daily convoluted process that happens continuously throughout our body's billions and billions of cells. Thank you for watching.